Thank you, and thank you for having me. And yes, I want to present today a new way of thinking about sex and its relations with the brain. And more specifically, I want to suggest that we move beyond listing sex differences in brain structure and function, and beyond thinking about brains as residing along a male-female continuum, to considering mosaic brains in a multidimensional space. And I want to start with what I think we all agree upon, and this is that sex, both sex chromosomes and sex hormones, affect brain structure and function in humans and animals, and that there are sex differences in brain structure and function. So I think we all agree on that. And the question I want to pose is, how do these differences add up in individual brains? Do they add up consistently, or do they mix up? And we know of at least one system where sex differences or sex effects add up consistently. And of course, this is the reproductive system. And I hope this will keep you awake for a while. So if you look at this great walls of external genitalia in men and women, what you can see is that although there is within sex variability, there is almost always internal consistency in the form of the different organs within an individual. So most babies are born with either only male genital organs or only female genital organs. And only rarely do we see a baby with intersex conditions because of having both male and female typical organs or an organ intermediate in form. So the question is, do sex differences or sex effects on the brain add up consistently as they do in the genitalia? And the reason to suspect that they don't, that they mix up rather than add up, is that we have quite a lot of evidence that sex effects on the brain, in contrast to sex effects on the genitalia, may be different, and in fact even opposite under different conditions. And not only that sex effects on the brain may be different under different conditions, these sex by other factors interactions can be different for different brain features. And this bulk of evidence together suggests that sex effects on the brain mix up rather than add up consistently. And because this is such an important principle, I want to demonstrate it looking at a single study, actually from Peg's lab. And what they've done here is look at the density of cannabinoid receptors in the rat hippocampus. And we can see a clear sex difference in both the dorsal and ventral hippocampus. Following 3 weeks of stress, we see that the effect of sex, or the sex difference, is reversed in the dorsal hippocampus. So what we thought was typical of males, this is high density, we now see in the stressed females, and what we thought was typical of females, we now see in the males. So this is an example of a reversal of sex effect or sex difference under different conditions. In the ventral hippocampus, something else is happening. So the interaction there between sex and stress is different. So the males, again, they switch from the male form, if you want, to the female form. So if we look here and we define the no stress condition as well, we look at the male typical and female typical form. So the males switch from a male hippocampus to a female hippocampus. But the females, they don't change here. So they switch from a female hippocampus to a mixed hippocampus. So they have a dorsal hippocampus in the male form and the ventral in the female form. So this is how the mosaic is formed. Now, there's lots of evidence of these type of interactions in many regions of the brain, not just the hippocampus, many regions of the cortex, amygdala, cerebellum, even the hypothalamus, for many different brain features, not just receptor density, but also dendritic morphology, size of specific nuclei, etc., and following many different manipulations, many involving stress, including stress experienced already in utero. So the mothers or the dams are stressed, and then you can see this type of reversal of sex effects in the adult offspring, and following other manipulations. Even simply housing the rats, one in a cage or several in a cage, can cause this reversal of sex effects. So if we go back to this single study, which tested the effect of a single manipulation on a single brain feature, and then we multiply it by the enormous complexity of the brain with all types of neurotransmitters and receptors and neurons, etc., etc., and then multiply again by the enormous complexity of the environment since the moment of conception until we all gathered here, then you can understand why it's hard to believe that brains will be internally consistent, and it's more likely that there will be mosaic of both male typical and female typical features. So this is a mosaic hypothesis which I formulated already in 2011, and it took me 4 years to understand how to test it in the human brain. And this is a study that I'll present shortly now. So what we did is look at MRI images of human brains. And we did many types of analysis with different types of MRI imaging techniques, etc. You can read it in the paper. I'm going to show you only one analysis, VBM analysis, for those who are more familiar. So we divided each brain into 116 regions of gray matter and assessed the volume of in each of these regions and created an Excel file, which I'm explaining because I'm going to use it. So each line represents a single brain, and it has 116 volumes of each of the regions. And we then colored each cell according to its size relative to all other brains. So if a region is relatively large, then we colored it in green. If it's relatively small, we colored it in yellow, etc. And when you do this to all of the brains, this is what you get. So on the left, you see brains of women, on the right, brains of men, and you immediately see the sex differences. So you don't need complex statistics to see the difference. There is more green on the female side, more yellow on the male side, and this is known. So when you use VBM, as we've done, women have, as a group, have more gray matter. This is why they have more green than men. So this is a really important figure for me because it shows at the same time that there are many group-level differences, but also that brains are neither yellow or green. Most brains are a unique composition or mosaic of green and yellow. So again, I want to stress that the question is not whether there are sex differences. You can see here that there are many sex differences and others, of course, and we've been talking the whole day about sex differences. So there are sex differences. I'm also not concerned at all with the question of the source of this question, nature or nurture, like Melissa described earlier. The only question that I'm interested in is whether these differences add up consistently within the brain of an individual, as sex differences in the genitalia do, or do they mix up, as we saw previously for the rat. And I think when you look at these images, it is quite clear that what is typical of both men and women is to have this mosaic of green, which is more typical of women, and yellow, which is more typical of men. So we can say that the typical brain types, or the brain types that are typical of women are also typical of men and vice versa. However, you can also see that there are some brain mosaics that are more common in one sex than in the other. So for example, yellow brains are quite rare in the population. But of the people that do have a yellow brain, there are more men than women. So the second part of the hypothesis is that there are sex or gender differences in rare brain types. And I want to contrast, or we contrasted this hypothesis with the hypothesis that it's very common that a typical male brain is different from the typical female brain. And just to give you one example of this hypothesis, which is often implicit, is not explicit, is from this study that probably many of you know of, the human connectome. And let's start with the image. So in the upper panel, all the connections that were stronger in men compared to women are depicted, and the lower panel depicts all the connections that were stronger in women compared to men. And the color means blue is for intra-hemispheric connections, and orange is for inter-hemispheric connections. And the way this figure is depicted, and the text that accompanies it, suggests that male brains are structured one way, and female brains are structured another way. Underlying this type of presentation and statement is the implicit assumption that sex differences add up consistently within each brain to create male typical brains that are different from female typical brains. However, according to our formulation, what's typical of the human brain, including in the connectome, would be to have brains, each a unique mosaic of both blue and orange. And we've shown this in the paper from 2015. So that's the question I want to contrast. Would the typical human brain be composed of both blue and orange, and actually these types of brains are extremely rare if they exist at all? Or is this a true representation of the typical male and female brains? Another example, of course, of the hypothesis that the typical female brain is different from the typical male brain is the extreme male brain hypothesis of autism from Baron Cohen. You cannot hypothesize that something is an extreme of something that does not exist. So if you assume that the autistic brain is an extreme male brain, you must assume that there is some, and this is from one of their papers, there is some normative phenotypic axis between a characteristic female and characteristic male phenotype. So this is quite a common hypothesis, the hypothesis that the typical female brain is different from the typical male brain. Now, I want to stress that in both formulations, there is acknowledgement of this. They start from the acknowledgement of the existence of differences at the level of single features. So these are actually two of the largest differences from this data set, showing that women have on average higher volume. I think this is a hippocampus and this is a caudate, but it's not really important. So in both formulations, there are sex differences in single features. The question is, do they add up consistently, as these images suggest, or do they mix up in individual brains? And before I go to present to you the study that we published last year about the entire brain, I just want to illustrate this question with a few of the largest known differences in humans between men and women, and this is from the lab of Dick Swab. So we have recently collaborated and reanalyzed his data, but this time looking at two features together. So you can see that in each of these measures, these are different measures of the INH 1 and 3, there is a large difference between men and women. It's about twice as many neurons in the men compared to the women in each of these figures. But when you look at this together, so when you take each two together, you see that there is no correlation between the number of neurons in each of these neurons. So you can have males that are very high on one measure but very low on the other, and vice versa. Okay, so again, the mixing up occurs even in features that show very large sex differences in the human brain. Okay, so back to this question. Are the typical male brains similar or different from the typical female brains? What we want to do, we want to move from the level of a single feature to considering the brain as a whole. And this is the actual representation of this data set. Each brain is a single data set. In the real world, each brain is a single point in a multidimensional space with 116 dimensions. Here we see only 2 dimensions, and these 2 dimensions are the 2 components of a PCA analysis that shows the largest sex differences, okay? And it's not really important for now, but what I want you to see is how your imagination changes when you look at the data, even in 2-dimension space, not in 116 dimensions. So when you look at a single feature, the typical male, different from the typical female form, is really very intuitive. And thinking about brain as residing along a male to female continuum, again, it makes a lot of sense. So does the extreme male or female brain hypothesis. But when you move to a multidimensional space, you can start to realize how this can also be true. So if you look here, you can see that most brains of both men and women are in the center, very dense center of the distribution. And there are some brains on the periphery. And when you look at the periphery, when only a few brains reside, you can see that some regions have more male brains than female brains. Other regions have more female brains than male brains. So you can start appreciating how this can be true you cannot understand how this can be true if you only have this image in mind. But once you move to a multidimensional space, you can see this packed center and the periphery all around, where you can see differences in the density of males and females. So the work I'm going to show you now has been published last year and has been done with collaboration from two mathematicians from Tel Aviv University. And all the analysis I'm going to show you have been done both in the original space, multidimensional space, and following different types of dimension reduction methods, both linear, like PCA, and non-linear methods. And you'll see it doesn't really matter. It was just to increase the generalizability, however you say it, of the conclusions. Okay, so the first thing we did was try to answer the question, are the brain types typical of women, also typical of men, and vice versa? And the rationale was this. We used an anomaly detection algorithm. And these algorithms were originally used to detect atypical computer activity. So if you want to protect your computer against viruses, worms, etc., you can use known viruses and protect against them. But you also want to protect the computer against unknown future viruses. So what the algorithm does, it learns your normal computer activity. And after it is trained on your normal computer activity, it runs in the background, monitors every computer activity, and for every computer activity, it tests whether it's normal or abnormal. And if it's abnormal, it will alert you, and maybe it's a virus and maybe not, but you'll get an alert that something abnormal is happening. So we use this algorithm, but we trained the algorithm on brains of one sex, so let's say females. So we took half of the brains of women and we gave it to the algorithm for training. And once the algorithm is trained, so it knows what a normal female brain is, we give it new brains of both women and men, and for every brain it needs to decide whether it's normal or abnormal. Now, if the typical female brain is the same as the typical male brain, you predict that the number of brains that are detected as normal would be the same, regardless of whether the algorithm was trained on male brains or female brains, because the typical human brain is the same for men and women. However, if, as others think, the typical male brain is different from the typical female brain, then you predict that if the algorithm was trained on women, then it will detect many more women as normal than men as normal. And of course, the other way around, if it were trained on men. And we did both. Just to make sure that the anomaly detection can really work and detect two populations if these exist in the data, we first tested it on the facial morphology of two primate species, and I have to thank my opposition, Marco Delgaidis, for this idea. And so I'm showing you the exact same thing that I'll do on brains in a minute. And what you can see here is that the results when the model is trained on macaques and when algorithm is trained on capuchin. And when the algorithm is trained on macaques, all the points fall on the y-axis. This means that it never thinks that capuchin is normal. It never thinks that a capuchin is a macaque. And when it is trained on capuchins, all the data points fall on the x-axis. So it never thinks that a macaque is a capuchin. Okay, so it makes no mistakes when there are really two populations in the data. What happens if you do the same with brains? So you can see that it's as opposite as could be. So it doesn't matter whether you train on men or on women. It doesn't matter which kind of dimension reduction method you use. All the points fall along the y equals x-axis. So the percent of males that are detected as normal is very similar to the percent of females that are detected as normal. This means that the brain types that are common in men are also common in women and vice versa. However, it says nothing about the brains that were detected as abnormal. And remember, the second part of my hypothesis is that there would be large sex differences there. So we use another method to answer this question. And we used unsupervised cluster analysis. So here we gave the algorithm all the brains, but with no information about the sex of the brain owner. And we asked the algorithm to cluster or to divide the brain into clusters, 2, 3, 4, up until 10, to best describe the variability in the population of human brains. And we used 2 algorithms, so each had a different criteria for what's a good way to divide the brains. And then after we get these 2 clusters, let's say these are the 2 clusters that the algorithm created, we look at the sex composition within each cluster. So we can assess how many males and how many females are in each cluster. Again, if the typical male brain is different from the typical female brain, we expect that large clusters will have one large cluster of mainly male brains and another large cluster of mainly female brains, and small cluster which whatever composition they have. However, if the common or the brain types common of men are also common in women, we expect that large clusters is what's common in humans, right? Because the algorithm doesn't know which is men and which is women. So large clusters are supposed to have a similar number of men and women, whereas small clusters, which represent rare brain types, may have large sex-gender differences. And again, we use 2 algorithms, the different methods of dimension reduction, and I'm not showing you all of this, you can look at the paper, only one type of presentation of the data. So what I'm showing you here is the proportion of men and women, whichever was larger, in a cluster as a function of the size of the cluster. And this is what you get. So it runs from 0.5, because there always at least half of one of the sex. So what you see is that indeed with the large clusters, the proportion of men and women is almost equal. So in common brain types, they have a similar number of men and women. However, in very small clusters, from 100 brains and less, you can see that the variability is very, very large. And you can get very high sex-gender differences. So you can have a cluster with 6 times more males from one sex over the other. Another thing that you can do following cluster analysis is you can calculate the chances the 2 men or 2 women or a man and a woman would be in the same cluster. And when you do this, you get that the chances are very similar. This means that knowing someone's sex category gives you very, very little information about the form of this person's brain. So knowing that Peg and me were both females gives you no information or no benefit of predicting how similar our brains would be than me and Greg, for example. Whereas, of course, if it would be in the reproductive system, you'll have more information that you ever wanted to know. So together, these 2 types of analysis demonstrate that the brain types that are typical of women are also typical of men, and vice versa, and sex-gender differences are found only in rare brain types. So, for example, if we think of autism, although we haven't tested this, you don't need to assume that the typical male brain is a little autistic to explain why there are more autistic men than women. The typical male brain is not more or less autistic than the typical female brain, but autism, which is a rare brain type, we don't know what it is exactly, but it's definitely rare, can have very large sex differences even without changing or affecting or making any assumptions on the typical male and female brains. Now, one of the greatest criticism or opposition to this statement, and also to our statement in 2015 that brains of men and women do not belong to distinct populations, was that you can use brain structure or function to predict the sex category of the brain's owner. And this is true. So if I know enough information about your brain, I can predict with about 80% chance to be correct whether you are male or female. It's not very, very interesting because I can just look at you and don't need to have you undergo MRI to predict your sex category. And if I'm not sure, I can ask you. But you can do it, if you really want to. But I want to illustrate or to demonstrate the difference between predicting someone's sex category on the basis of someone's brain mosaic and predicting someone's brain mosaic on the basis of someone's sex category. And this is just an illustration of this argument. And to do this, I want to move for a minute to another domain, and this is text, writing, books, etc. And researchers from Bar-Ilan University have shown that you can predict the sex of the author of a text on the basis of known sex gender differences in writing style. And what you need to do is you need to count the number of occurrences of words like the, a, some, etc. You have it, and you put it into this formula. And then if the product of this formula, or the number that you get, is smaller than the total number of words in the text, then the author is most likely a female. If it's greater, then it's most likely a male. And you have about 80% chance here to be correct. So this is how you can use gender differences in the text to predict an author sex. You can also do the reverse. So if you know that an author is, for example, a female, you can predict that there is 80% chance that the product of this formula would be less than the total number of words in the text. Okay. Does this has any meaning, this prediction? It has no meaning. It doesn't even tell you the number of each of these types of words. There are endless number of solutions to this, to what I just said. Moreover, and this is why I like this example, if I asked you to divide books into categories, I guess this would not be your first category. You would probably divide books according maybe to length, genre, whether this is science books or fiction books, maybe if it's interesting or boring. So there are many, many variables along which you can create categories of books. So going now back to the question of the prediction in the brain. The fact that the true observation that you can predict one sex category on the basis of brain structure or function does not necessarily, and I stress, does not necessarily mean that sex category is an important variable in accounting for the variability in brain structure or function. Just as the fact that you can predict the sex of an author does not mean that the sex of an author is an important variable in accounting for the variability in books. It could be an important variable. It doesn't prove that it's not, but it's not a proof that it is important. And this is what we've seen here. So the algorithms were able to create divisions or clusters of brains. These clusters were not according to sex. So there are other variables which we don't know of yet, maybe we should start studying them, but there are other variables that account for much more variability in brains. Whether this is meaningful functionally or not, we don't know. We didn't test for the functional meaning of these clusters. Okay? So there are other variables that account for much more variability in brain structure than sex category. And I leave it to you to decide whether sex category is important or not. But so the first point is that the fact that you can predict sex category doesn't mean it's important. And the other point is that the fact that you can predict sex category doesn't mean that the reverse prediction is meaningful. And I want to illustrate this looking at brains. So for example, if I tell you about 10 features that show large sex differences in the human brain, that this feature is in the male typical form and this feature is in the female typical form. Okay, so I give you all the what's male typical and what's female typical and what's intermediate. And I ask you to guess the sex of this author. Probably you would guess that this one is female, right? And what would you guess for this one? Female. And this one? Also female. So the rule that you are using is that if a person has more female characteristics than male characteristics, it's probably a female, and the other way around for males. And if you are using this type of very, very simple criterion, you will be right in about 80% of the time in classifying brains or guessing the sex of the brain owner. But the question is the opposite. Okay? I know someone is female, what can I say about her brain? And the only thing I can say about the brain is that there is 80% chance that her brain has more female characteristics than male characteristics. This is some information, but not a lot. It could be 1 and 0. It could be 10 and 0, it could be 5 and 4. And it's not just that I don't know the number, how many male typical and how many female typical features, I also don't know the composition. And of course, the composition is what is important about function. So if you take this brain, which is obviously female because it has 4 female typical features and 3 male typical features, and you compare to this, another female typical brain if you want, then these brains are almost as opposite or as different as 2 brains can be. Whereas this brain is very similar to the first brain, although it is a male typical in terms of the number of male typical and female typical features. So, and again, I remind you that when we use unsupervised clustering, the chances that a man and a woman would be in the same cluster, that is, will have the same brain type, is the same as the chances that 2 men or 2 women will. Okay, so we started with the question of there are sex differences, sex affects the brain, how do these differences add up in single brains? And I hope to have convinced you that they do not add up consistently as sex effects in the genitalia do, or as these images suggest that they do in the brain. But rather they mix up in such a way that what is typical of human brains is to be a mosaic, each a unique mosaic of male typical and female typical features. And the reason that the differences are mixing up, and this is important, is something about sex itself. It's not because of gender, etc. It's because how sex affects the brain can be opposite under different conditions, and because these interactions are different for different brain characteristics. These 2 observations are the reason for this mixing up of features or sex effects within each brain. And some people ask, so maybe what's male or typical about the brain is not the structure of the brain as we see here, maybe it is how it responds to the environment. For example, how it responds to stress. So maybe what's male about a brain is the way it responds to stress. And my answer is that no, it will not work, because how stress affects the brain in males and females depend on other factors in complex ways. And just to give you, I don't have much time, so just to give you the idea. So for example, this is the behavioral effects of exposure of rats to stress in adulthood. And you can see that these effects are different if the rats were also exposed to stress when they were in young age. However, this type of augmentation of the early stress, of the effects of the late stress, are different in different regions. So if you look at avoidance, there is no effect. And if you look at escape, there is effect in males, but not in females. So how stress affects the brain of a male or a female depends on other factors. So also there you will get a mosaic. And we've recently tested it in the human brain. This is not published yet. And what you can see here is we looked at the brains of soldiers before and after military service, and they underwent very bad experiences during the military service. One soldier was actually killed. So very, very stressful experiences. And what you see here is 7 brain regions in which the typical male and female response to this stress was different. And the color is, if it's pink, then this is a female typical response, and blue is a male typical response. And so each line is a single brain of a single soldier, women here and men here. And again, you see the group level sex differences. This is how we define what's pink and what's blue. But you can also see that there was only one soldier that was internally consistent in response to stress, and about 2/3 of the soldiers had both male typical and female typical responses to stress. So also here we are mosaic of features. Okay, so if you go with me and we start to think about mosaic brains in a multidimensional space, what does it mean for research? And I think the first thing is that we should always use males and females in research, in animals and humans. And this is because we want to capture the entire variability of the species. If we use only one sex category, we don't. The question then becomes, should we use sex category as a variable when analyzing the result? And now the NIH is requiring that we do. And I'm not sure it's a good idea that we do. So I want to answer this question, but to separate it for discussing humans and animals. And let's start with animals. So when we are using laboratory animals, yes, we should use sex category to analyze the results, at least in the first step. And this is because the environment is highly controlled. We control the strain we're using, so the genetic background is quite homogeneous. We control the pregnancy, we control how they grow up. So the environment is quite controlled, and therefore we are more likely to see the effects of sex on these animals. However, this means that whatever we are measuring is true for this specific strain, conditions, etc. And we should always keep this in mind, that had the conditions been different, we could have gotten very, very different results. So yes, we can study the effects of sex there, but it is the effects of sex under these specific conditions. The other thing that we need to remember is that we only, when we measure sex or when we compare males and females, sex is a proxy for many, many variables that may be causing this difference that we are observing. And this is from a paper that Peg and I wrote, and I won't go over this, but a difference between males and females in an endpoint can be a result of the effects of sex chromosomes or sex hormones. But it can be the result of many other factors that correlate with sex category, from housing conditions to weight, etc. And even more important, when we compare males and females, we are comparing individuals that there is a lot of overlap between the different measures. Probably many are mosaic, so an animal or a human can be high in testosterone, but still very short in height, for example. So we are not comparing two groups, each receiving a different drug. We are comparing groups that are highly overlapping, and each individual is a unique mosaic of all these sex-related factors. So yes, when we study animals, we should, or we could use sex as a category to analyze the data, but finding a sex difference is only a first step in the study, whereas today, usually it's a last step. So we found a sex difference, and that's it. It's easier to publish, but we know nothing about the mechanism. When we go to human studies, then my answer is that if you study the normal human brain, then you should not use sex category as a variable, because it will only yield false positive results. And if you think about, you have in mind this image, then when you take two samples, 20 men and 20 women, and you put them in the MRI, you think that what you're doing is taking two samples from two different populations. But what I've just shown you is that you are actually taking two samples from a single highly heterogeneous population. So you are likely to find differences, but this would be false positives. And there was recently, a paper was recently published, which I wish I have done this, but someone else did. And what they have done is the following rationale. If brains of men and women indeed belong to two different categories, then the larger the sample, the more likely we are to find differences, right? This is basic statistics. So what they did is look at the number of differences in functional MRI studies in humans as a function of the sample size. And there is no such relation. So again, if brains of men and women were belonging to two categories, to two populations, you would expect that the larger the sample, the more differences you find. This is not the case. There is no correlation between the two. So again, this supports my claim that we belong to a single highly heterogeneous population, and it would be just a waste of time and energy to use sex as a variable there. Okay, so I end even on time, and I want to thank you for listening.